fills up. Okay, we're recording.
Okay. Time is it? We have 2 minutes. 1 minute.
Yeah, a couple of minutes still.
Okay. Apologize for the bad lighting. I know what we can do. Let's do it this way.
You're back in presenter view, Alan.
There, how's that?
I think that would work. It's not full screen, but
Oh, all right.
Still in presenter view, Alan.
yeah, you might want to switch displays again.
Just trying to get better lighting of me there, because I hate it when people are all silhouetted. So I just had to unplug my display. And now it's wrong way around?
I think so. I think you're just blown up because I see the little next slide and the
Yeah,
circle
yeah.
on the top. So if you switch displays again, it should give us the.
I'm going to go back out, back into, oh, I'm going to stop share and then share.
Okay.
I think it got messed up. Okay, you can,
Okay, there we go.
all right, sorry.
All right, so it's time to get started. Welcome to seminar, everyone. The first thing I'd ask is if you can each mute your microphone so we don't have interference. Thank you. And today for our seminar speaker, we have our very own Dr. Alan Miller, who's going to talk about reprogramming protein synthesis by SARS-CoV-2. And in case you don't know or don't remember what that is, I'm sure Alan will remind you of what this virus is, and it's very appropriate for this time. So with that, I'll let Alan get started and look forward to hearing about this.
Okay, thanks, Leonore. Yes, well, Leonore told me that everybody knows that SARS-CoV-2 is the causal agent of COVID-19, right? So SARS-CoV-2 is severe acute respiratory syndrome, coronavirus 2, and it causes coronavirus disease 2019, which is COVID-19. And this is the, this talk is going to emphasize the microbiology part of plant pathology and microbiology for today's seminar. And so I'm going to start with some artwork to show how what a pandemic of a respiratory virus can make you feel like. So this is Edvard Munch, a self-portrait. So he's, you might know him for The Scream, a very popular, rather disturbing painting. So he did self-portraits of himself suffering from the flu on the left, and you can see he's very pallid and can hardly breathe. And I think this is probably not a lot different from what a lot of COVID-19 sufferers feel like. And then the picture at the right, believe it or not, is a self-portrait after he recovered. So you can see at least he got his color back, and but he still doesn't look great. And then another artist who I think sort of portends the situation now is one of my favorite American artists, Edward Hopper, and he captured a lot of different sort of people have noticed this, have seen, and including this. This, by the way, this art is from an article in Cell, so it must be science. But anyway, you can see the loneliness of this woman just staring out her window. And I don't know about you, I actually have had to quarantine myself, and plus we had lockdown, and I did a lot of window gazing just like her. And then he also predicted the social distancing. You can see these kind of cold, isolated commuters in the kind of luxury car of a train in the 1960s. The only thing they're missing is masks. So I know a lot of us are kind of feeling this way now, thanks to this virus. And I also thought I'd give an update of why, we all know why it's important to work on this virus. It's already caused trillions of dollars of economic losses. But this is a website that I recommend. There's a lot out there. This is called globalepidemics.org, and it's from the Brown University School of Public Health. And it's a really good interactive maps of where you can click on any county, any state, or any country and see how many cases were reported in the last week with the number of cases per 100,000. And so right now, it's pretty depressing. This shows a statewide map, and the US average right now is 21 cases per 100,000. And this color coding is shown down on the lower left. So red, which is what most of America is now, geographically at least, is 25 or more cases per 100,000. And that is called the tipping point where stay-at-home orders are necessary. I don't see a whole lot of that. Now, maybe if we had 80, 90, 100% mask use, we wouldn't need this. And then orange is 10 to 24 cases where contact tracing and stay-at-home orders are recommended. Then the yellow is community spread, contact tracing recommended, and green is things are looking good. So there's not a single state that is green. We zoom in on Iowa, you can see the whole state is almost red. We can take a little very, very slim satisfaction. Story County is just barely in the orange because we have 23.5 cases per 100,000 right now, which is slightly under 25. And that's way better than Labor Day weekend when all the students came back and we had that big surge. We were up at 110 positives per 100,000, but it's worse than a week ago. So we are like everybody else trending in the wrong direction. So I could go on, but I'll just focus on the subject of my talk, which is how one aspect of how the SARS-CoV-2 virus controls its gene expression. And we're studying ribosomal frameshifting. I couldn't resist getting into this because we basically had all the tools in my lab. We studied ribosomal frameshifting of barley yellow dwarf virus from in the 1990s and early 2000s. And so I knew about the coronaviruses because they use the same mechanism, and it's pretty much the same mechanism, and it's the same tools to study. They have different RNA structures, but really the only thing different that we had to do is get a hold of some mammalian cells to study gene expression instead of plant cells. So to do that, I got together with Kathy Miller, who's a human virologist. She studies RNA viruses out at the vet school, and also with Walter Moss. He's an assistant professor in BBMB, and he's a real expert in computational prediction of RNA structure. And he's actually done lab work as well using RNA-seq techniques to look at RNA structure in vivo. And he's got some virus experience, so he was very excited about this project. So we got together and we got a small grant from the Office of the Vice President for Research at Iowa State. they gave out several little awards so that people could kind of drop everything and start using their expertise to work on this COVID problem. And I'll give a little introduction to the classification of the virus. I found this kind of interesting. So this is the official classification from the International Committee on the Taxonomy of Viruses that was published a few months ago. And it's new to a lot of virologists because the virology classification is really changing fast these days. So it's an RNA virus, which puts it in a realm called riboviria. And it's a positive strand RNA virus, I should point out, which is what most viruses are. Most human viruses and definitely most plant viruses are positive strand RNA viruses. We'll get into that more in a moment. And anyway, the family is Coronaviridae, so that's why it's a coronavirus. And for comparison, they show how humans are classified. So we are in the order primates, the family Hominidae, the genus Homo, and SARS-CoV-2 is in the genus beta coronavirus. Then it gets kind of interesting down here. The species is not the same as the virus, which I only learned recently for viruses. Just a few years ago, I learned this. So it's severe acute respiratory syndrome related coronavirus. And then they list examples, and these are considered not strains, but actually like individuals. For comparison, you compare with individual human beings. And here's some plant pathology for you. Some of you may notice some couple famous plant virologists are listed among this list of random human beings who lived in the last century, basically. Anyway, SARS-CoV-2 highlighted here, the Wuhan strain, I'm still going to call it that. That's like the type isolate, is actually considered the same species as the SARS-CoV that where we had an outbreak in 2003. Even though that one was very different in its epidemiology, it had a 10% case fatality rate, much more dangerous to get, but much less, it did not spread anywhere near as readily as SARS-CoV-2 does. And then here's a phylogenetic tree to show the relationships of the ones that are infect humans, and they're considered zoonotic viruses, so they're considered animal viruses that got into people. And SARS-CoV is actually only 79% identical in nucleotide sequence. So I'm really surprised that it's considered the same species as SARS-CoV-2. Normally with viruses, those would be different species. And then there's another virus some of you may know about, the MERS virus, Middle East Respiratory Syndrome. This is actually a camel virus that occasionally gets into humans. It's not very, it doesn't spread well among humans, but if you get it, it's really bad. It's got like a 33% case fatality rate. And it's maintained at a low level in the Middle East, but I think if you avoid camels, you're pretty much okay. And then it's important to point out these ones in orange. These are a little more distantly related, and these are common cold viruses that we have gotten forever. They've always been known about. And a concern is with these viruses, one reason why we get colds all the time is when it comes to coronaviruses, you can get reinfected by the same coronavirus again. So it doesn't bode well for antibodies lasting a long time. And that's been seen with SARS as well. So when it comes to a vaccine, it may mean that we have to get revaccinated every year, for example. So moving on to virus gene expression now. So there's a positive strand RNA virus, as I said, where the mRNA is the genome. And the gene expression is a bit different. So to remind some of you, getting all the way back to the central dogma of molecular biology, as you know, in organisms, non-viral organisms, they have a DNA genome, and the vast majority of gene expression during development or in response to environmental changes is at the level of transcription. So DNA making the mRNA. So we have all sorts of different promoters on different genes and enhancers. And then there are all sorts of transcription factors that control the bulk of the gene expression. And many viruses, such as HIV, exploit that. So HIV is an RNA genome, but it quickly gets converted to DNA that integrates. And then it's regulated in many ways using the host nuclear transcription and splicing and nuclear export machinery to regulate its gene expression. Some viral proteins influence this though. While positive strand RNA viruses, which as I mentioned are the most common kind of viruses, they don't use any of this. They can't. They don't even go in the nucleus. So they have no transcriptional control at the DNA level, no splicing. Their whole lives are lived in the cytoplasm. They do build their own little organelles in the cytoplasm for replication. So that makes translation a much more important step for gene expression control, which is why I've been studying RNA virus translation most of my career at Iowa State. So getting to the actual viral genome. So here is the SARS-CoV-2 genome. As you can see, it's a huge RNA molecule. It's one of the biggest RNA molecules known. It's basically almost 30 kilobases, a single RNA, and it's a single messenger RNA. But, and the genome itself serves as the message for translation of these huge open reading frames. So this open reading frame 1A, it codes for, it is 13,000 bases long. So you get a single protein made from that, but you'll see it's not really a single protein. And then ORF1b is also translated from this by, it requires this ribosomal frameshift event, which will be the subject of my talk. So that's 2/3 of the genome. Now the rest of the genome, encoding all these genes down at the 3' third of the genome, those are not translated from the genomic RNA. During the course of virus replication, subgenomic mRNAs are generated. And so each one of these serves as a message for pretty conventional translation, it seems. They have a 5' cap, they have a poly A tail, they all have the same 3' end as the viral genome. And the first open reading frame on each one of these, there are some exceptions that aren't shown, but the first open reading frame is what gets translated. So it's pretty normal translation there. And this includes what we call the structural proteins. So the proteins that are actually in the virus particle. And that's shown over here. So the red squiggly line over here, that's the viral genomic RNA. The other RNAs, these subgenomic mRNAs, do not get packaged. So when the virus first starts to infect and the particle enters the cell and the RNA is released, the only message in the cell is the genomic RNA. So these are expressed a little later in infection. That's one way it regulates gene expression. But it includes the proteins, the nucleocapsid protein, highly abundant. It coats the RNA and it's all stuffed in a particle. There really is no empty space as is shown. And the particles are more blobby, like I showed on my opening title slide. There's a membrane envelope around the particle that has membrane proteins. And then the famous spike protein that's being used for all the vaccines, that's this big green protein on the surface. And all these other viral proteins, including all the replication proteins that are made from ORFs 1A and 1B, those are not in the particle, they're not packaged. So the virus, when the RNA enters the cell, it cannot replicate until it first translates. So that's one reason why I've always thought translation is a good target to nip virus replication in the bud for antiviral approaches. So to look at the actual proteins that are translated from here. So as I mentioned, you get this big protein called PP1A. It's called a polyprotein. And then you also get this PPA1B made by frameshifting, which I'll go into in a moment. But these are polyproteins, which means actually really different proteins are all translated together, and then they're proteolytically cleaved apart to be the functional proteins. So it really isn't like a giant protein is made. And some of this cleavage probably occurs co-translationally. So these really large proteins may not really exist in the cell at one time. And included in the functions of these are the actual proteases themselves. So this PL-Pro here and this 3CL-Pro, NSP5, they cleave themselves out. They cleave at these specific amino acid sequences indicated by the triangles. And then the timing of the cleavage is such that it's not all at once. So sometimes you'll have several, some cleavages will be slow. So you'll get a sort of a mini polyprotein and that'll have a certain function for a while. And then it'll get fully cleaved. And those fully cleaved products might have a somewhat different function. So it's kind of a post-translational way of controlling gene expression through proteolytic cleavage. And this is common. I think all positive strand RNA viruses of vertebrates use this. Not all plant viruses. Some plant viruses do, but I teach virology. I was just thinking about it. I think they all have some aspect of polyprotein in them. Then there's this frameshift to make, so you get this super big polyprotein, and that allows translation of, crucially, the RNA-dependent RNA polymerase. So that's the key replication enzyme. And that can only be translated if there's frameshifting. And that same enzyme that applies to Barley-Ehlendorf virus. And it also applies for translation of the reverse transcriptase of HIV. They all use frameshifting. Not all viruses, but those three do, and others. And then there's other replication-related enzymes, including an endonuclease and an exonuclease, and a helicase to help melt out RNA structure. And the nucleases are involved in proofreading. So if there's an error in base incorporation, it'll get cut out by the exonuclease, and then it'll be copied again by the polymerase. And only coronaviruses, they're the only RNA viruses that have this proofreading. The others just make lots of mistakes, and it helps them evolve. But this genome is too big. If it made lots of mistakes, it would never have a viable virus. So it has to have higher fidelity replication. Okay. So how does that frame shifting work? And okay, the frame shifting occurs not all the time. So most of the ribosomes, when they reach this stop codon, they stop and they're done. But some will do the shift, which allows them to keep going. And only about 20 to 30% do that. Or here I'm saying 20 to 25%. So that means you get only about 1/5 or 1/4 as much of these proteins made as the upstream ones. So it's a regulatory mechanism to adjust the ratios of proteins. And then, yeah, it's just an amazing RNA with a huge non-coding region because these don't get translated from the genome. And that's a whole other story because most mRNAs with huge 3' UTRs are destined for degradation. So how does it work? Well, ribosomes initiate pretty conventionally as far as we know. And then, as I said, most of them hit the stop codon and stop, but a few shift and translate ORF1b. So what induces this shift? Well, my colleague Ian Brierley at Cambridge University has been studying this in a distantly related coronavirus called infectious bronchitis virus. It's actually a chicken virus. And he started studying that about the same time I was studying DYDV. So we followed each other's work. And this same type of structure is required for frame shifting for both in general principles. So what you have is, I'm zooming in now on the actual sequence where the frame shift occurs. So you have the shifty site or slippery site. In this case, it's UUUAAAC in green. And so what happens is ORF1A is being translated, and in that frame you have UUA encoding leucine, AAC encoding aspartate. And then the tRNAs that bring in the leucine and the aspartate, so they're anticodons, their triplets are base paired here. And then the tRNAs and the whole ribosome will shift back one base and start translating again. So they would be base paired with a mismatch to UUU and AAA. Then the next amino acid incorporated would be arginine, coded for by CGG and then GGU for glycine. And then they keep going to translate ORF1b. And then the other 75% or so of the ribosomes, they don't shift, they just keep going. So they do leucine, aspartate, glycine, valine, and pretty soon they hit a stop codon and they're done. So that's the guts of the frame shift region. And then what makes it shift? Because UUUAAC probably occurs all the time. So you also need downstream at a very specific spacing, a very structured region. And it's usually a pseudoknot like that's shown here. So a pseudoknot is when the RNA, it base pairs to itself, which is very common, and it's forming what we call a stem loop. So you have the helical region, and this loop goes CA, and it comes down GG, so on, comes down like this. So that's the stem loop, but then you keep going downstream, and it loops back and then base pairs to itself in the stem. And it's And that's what the pseudoknot is. And it's drawn this way because helical regions or stems, they like to coaxially stack on each other and they can be very stable. So the idea is thought that the front of the ribosome is bumping into this pseudoknot and it has a hard time resolving that structure. It eventually does, but 25% of the ribosomes, before they do that, they sort of bounce back like a spring almost, maybe. They get pushed back a base. So that's what's thought to cause the frame shift. Okay, and then Ian Brierley actually did some cryo-EM to show this in the ribosome. So the gray is the ribosome, and then where the mRNA enters inside the ribosome, this pseudoknot kind of gets stuck. You know, it binds or interacts with flanking ribosomal RNAs and proteins to slow things down a bit. Yeah, so not any pseudoknot will work though. So there's very certain structural features. You can engineer a even more stable structure and ribosomes might have no problem getting through that. So there's the specificity of interacting with the ribosome. So what about SARS-CoV-2? It is, like I said, it's in a different genus from the virus I just showed. And its pseudoknot is fairly well understood already because in this part of the genome, it is virtually identical to the original SARS-CoV virus. And so that has been studied since 2003. And what's been found so far is it has the standard shift site and a spacer. And then here's the pseudoknot, and I drew it in a different way to kind of show the stem loop, the loop part a little more clearly. So you have this stem one and then a loop, and then the part that base pairs downstream is here. And in this case, there's actually a mismatched base in the middle. So there's a little bulged out base. And then there's also this whole extra stem loop that's just inserted in here that is not present in the other virus. And so I drew it another way on the right, which looks a little more the way I showed it on the previous slide, where stem 1 and stem 2 are both kind of vertically stacked. And then this stem loop is out here. And there's only one base difference between, in this core region, this is like the core frameshift element. This will give you frameshifting, but it may not tell the full story. And we're actually interested in how flanking sequences might affect this. But this A is a C in SARS-CoV. So that's the only difference within this region. And we do a lot of predicting structures using various computer algorithms, including Walter Moss's program, Scanfold. But you still have to do a lot of sequence staring and looking at alternative possible base pairings. Pseudoknots are really hard to find by computer. So it reminds me of when I play this game Boggle. I don't know how many of you play Boggle, but I think it's a really good exercise for gazing at RNA structures and trying to find things like pseudoknots, because you get a random set of letters in on these cubes. And from here, you have to look in all different directions to find words. So you have rules, like they have to be English language words that you can find, like M-A-N-E or something. And here we're doing, it's very similar kind of parts of the brain that function here, I think. I just thought I'd throw that in there. So how do we assay this? So what we do is we use a dual luciferase reporter. Basically, it's designed so we have an upstream reporter gene, a Ranilla luciferase, which comes from jellyfish. And then we have it arranged so that translation of the firefly luciferase, which is from fireflies, this firefly can only be expressed if the -1 frameshift occurs. So what we do is we design it that way. So we insert the viral sequence shown here in such a way that the Ranilla will be translated, and then we remove the stop codon. So the protein is fused to the viral sequence. We go through that 3-helix pseudoknot, and then we start translating the firefly sequence. So we make sure we don't have a start codon here. We don't want any internal initiation. So again, the only way the firefly is translated is by frame shifting on the viral sequence. So a few viral amino acids are fused to these. And then to measure the efficiency of frame shifting, what we do is we just insert one base somewhere. And if you think about it, if you sit there and start counting the triplets, that makes the firefly be in the same reading frame as the Ranilla. So now when we translate, vanilla and firefly are all one, then no frame shifting needed. So that's sort of what you would get if you had 100% frame shifting. So then we assay both constructs, and then we just take the ratio. How much firefly did we get that required -1 frame shifting? Divide that by the amount of luciferase activity we get in the in-frame construct. And I should mention, these are really easy to assay enzymes. There's kits for this. There are actually high throughput drug screens, like for testing thousands of drugs that affect gene expression that rely on these dual luciferase reporters. So and we already were working with it in the lab for a whole different reason. So all we had to do to shift from plant virus translation to SARS is synthesize this sequence I'm showing here and plug it into the reporters, and then get a hold of some mammalian translation system. So to confirm that it works, that we get frame shifting, I forgot to mention, I'll back up. Then this is all in a puck-type plasmid, and what we do is to make the mRNA, we have a bacteriophage T7 promoter here, and we linearize the RNA at the 3' end right at the poly A tail, and then we in vitro transcribe the entire RNA to plug it into our translation systems. And the easiest ones to use and the very informative ones are the in vitro ones, the cell-free extracts from rabbit reticulocytes or from wheat germ. These are commercially available. Rabbit is good because it's a mammal like humans are, the host of SARS-CoV-2. Wheat germ is good because it's a very high fidelity system. And so far, it seems like all the rules for frame shifting are obeyed by wheat germ. So plant and animal ribosomes and elongation factors are so similar that they'll behave the same for translation of a mammalian RNA versus a plant RNA. And so this shows the first experiment to sort of verify that what we see with our luciferase readings are actual, what we expect. This was done by Liz Carino, who many of you know. She got her PhD in May, and then she stuck around for a few months to work on this project. So this shows the translation products, the radio-labeled methionine of the RNAs I just showed. We have a negative control that I didn't mention, where you just put in a bunch of stop codons after the ranilla. So this shows the mobility of ranilla luciferase by itself. This is the wild type, or the one that requires -1 frame shift. So you see ranilla made by the ribosomes that chose to stop. And then you see the fusion of ranilla and firefly up here, the higher molecular weight. Those are the results of the ribosomes that frame shifted. And this one is 3 times bigger protein, so it has probably 3 times as many methionines. So on a molar basis, this band is 3 times darker than this one. So really there's, on a molar basis, there's about 1/3 as much protein here as down here, about 30% frame shift. And then this is our in-frame control. We have the same thing with uglier gels because reticulocytes just aren't as clean as wheat germ over here on the left. And then moving on to measure the actual luciferase activities and calculate frame shift ratios from that. We see on the right, we see results of repeated experiments. The first 2 are the reticulocytes again in the wheat germ. So it's basically the same as on the left, but now we're measuring luciferase activity. The top is the in-frame control luciferase, just normalized as 1. And then the levels we see from retics and wheat germ. So it's about 32 and 26% frame shifting. Then on the left are the more biologically relevant ones, you could argue. This is where we look at frame shifting in cells, in mammalian cells. And we used Vero E6 cells shown here. These are monkey cells, which are the most popular cell line used now to actually study SARS-CoV-2 replication. So we know the virus replicates in these cells. And this work was done by Luc Boussière and Kathy Miller's lab. He transfected the RNA into the cells, and it's kind of a slow process. And we don't exactly know how much goes in, but it's probably the closest to mimicking what the virus does. The virus gets taken up into the cytoplasm, and there's probably very few RNAs to initiate the viral infection. So this is very similar to that. And we're actually seeing only 10% frame shifting. But again, these numbers we can't really extrapolate to the virus because it's only about 100+ bases of viral sequence in a reporter construct out of context. And we see a lot higher frame shifting here in these baby hamster kidney cells. These are expressed in a different way. Here we actually have T7 polymerase engineered into the cells. So we just transfect with DNA plasmid, and then the polymerase inside the cell in the cytoplasm transcribes the RNA. It makes just tons of RNA. So this is blasting the ribosomes with tons of RNA. And in vitro, we have ribosomes that have no mRNAs on them. They're just looking for something to translate. So it's a very translation, really favors the added RNA translation. And in vivo, you have the cells are already busy, ribosomes are all busy translating the host mRNAs, and the added RNAs have to compete. So that might explain some of these frameshift differences. But the important thing is we want to look at is the effect of mutations and to determine the structure. So what we have here are the predicted structure on the left that I mentioned before, but Walter Moss's lab, when they were looking at the structure of the entire genome, they predicted that upstream of that region, there's more base pairing. For example, the shifty site itself looks like it's in a little stem loop. There's this stem loop here called an attenuator because in SARS, a stem loop in this location caused a reduction in frameshift efficiency, or so it was reported. And there, the sequence is actually quite different from this stem loop. So the core region's the same, but upstream is different. And then there's some base pairing here that is predicted by Walter's algorithm. And then I found by looking closer that that base pairing could extend up where it would compete. So this sequence in blue could base pair to the sequence in green, which would disrupt the stem. And if we extend further upstream, we get something like that. So these are the same sequence and the colors are the same to help you sort of line things up. But the sequence on the right definitely would not frameshift. There's no pseudoknot. And so we designed some experiments and we're still in progress. This is definitely an in progress talk here to try to figure out if this structure, if this base pairing, especially this blue region, can compete. And I'll show some of that on the next slide. So here's some actual data where we plugged this sequence. Unfortunately, we only had part of the competing stem. We plugged it into the dual luciferase reporter, and then we look at the frameshift activity in all 4 types of translation extracts. We made a bunch of mutations. For example, we changed this CGC to a GCG, so it's the same sequence as this, so it should not base pair to that, and it shouldn't compete. We thought maybe this will increase the frameshift rate because it'll favor formation of this stem. But really didn't see much. It looks maybe a little higher, but it's not really reproducible. Then we made a different mutation, M2, where we changed the GCG to CGC. So it can't base pair to this, nor can it base pair here. We shortened this stem, and it really knocked down frameshifting. And then we did the reciprocal mutation where we changed the other side instead, same result. And then we made a mutant that is a little surprising, where we combined these 2 mutations, so only the competing stem should form and not the bottom of the green stem needed for frameshifting. And surprisingly, we got slightly higher frameshifting. We thought this would be dead, but that's why we do science. We get unexpected results. And then satisfyingly, we got a very expected result with this mutation, which is combining these 2 mutations. So when we mutate to make M2 or M3, they disrupt the stem. We combine the 2 to restore the base pairing with the sequence flipped. We restore frameshifting. So that shows pretty much proof that the bottom of this stem exists and is needed for frameshifting. I should point out that in the natural sequence, there's a GU pair, which is a weak pair, but a legit pair. We replaced it with a stronger CG pair. And we made more mutations, like M7 is a complete flip of all 3 sequences. So we have a GCG, CGC, GCG, and we get pretty much on average wild type frameshifting, maybe a little lower. And then M8 is we switched it, we only made 1 base change. Here's M8. We turned it into the SARS sequence, the original one, and it's a little scattered here. I don't know why the wheat germ is so high, but again, we and other labs have already shown this, that we think both these frame shift at the same rate when we're focusing only on this core region. When we move out, and oh, I forgot to mention, just to confirm that these luciferase results are made by frame shifting. If we run the products on a gel, we can see exactly the same results. So M2 and M3 make very little frame shift product. M5, we restore the frame shift product, for example. Okay. And that the gels were done by Liz, and all these luciferase assays here were done either by Liz, the in vitro ones, and the in vivo ones were done by Luc Boussier. Now moving on to that attenuator stem loop I mentioned. I think I'm getting behind on time. So I'll just quickly say we did the same type of mutagenesis where we wanted to see what effect this orange stem loop here is having, if any, on frame shifting, as it was reported to for SARS. Remember, this is a different sequence than SARS. So we introduced mutations in one side of the stem, M9, these ones in gray, or on the other side of the stem, in M10. And as you can see, they're predicted to totally disrupt that stem region. Then we combine them back together. It restores base pairing with the GC and CG here flipped. And the stability measured by the delta G. And the more negative it is, the more stable the base pairing. They're very similar, right? But when we look at the results, M9, M10, M11, the results are here. In vitro, there's no difference. In vivo, we see, for some reason in these Vero cells, we seem to see less frame shifting in all of them, not more. And the double mutant didn't restore it to wild type activity. So it's not what we predicted. And then when we look at the SARS, we replaced the whole thing with the original SARS attenuator. So this for sure should attenuate if the previous paper was right. We don't really see that. And then just for the heck of it, we decided to make a really stable one if you look over here on the left, we have a UU mismatch. We zipped that up. We changed a U to an A. So now it's a very stable stem, totally unnatural. And there in vivo, we did get a reduction in frame shifting. So maybe a stem loop here has the potential to reduce it, but we don't see it in any natural constructs. And then, okay, so moving on now, we wanted to see how much more time do I have, if any?
Seminar ends at 4:10, so Alan, you still have 10 minutes.
Okay, all right, good. So I wanted to show some of the BYDB work we did. Back in 2002, we were looking at frame shifting of this little small genome compared to SARS-CoV. And here's where the frame shift site is on the lower right. And it's required again to make the viral RNA-dependent RNA polymerase. We translate this in vitro. This is not a reporter assay. We're translating the whole genome. In this case, we did use the reporters a lot. You can see the product of the first open reading frame and then the frameshift product, and there's hardly any because this is much less efficient. This is like 1 or 2% frame shifting. So it's actually a lot easier to work with the SARS-CoV. It's quite exciting to do these experiments because you get so much frameshift product. But this one, we got some structure here. We didn't get any frame shifting unless we had a sequence way down here, 4,000 bases downstream. And the reason why is here's the pseudoknot. It's a different type of pseudoknot. Here's the shifty site, here's the spacer, here's the structure. And then there's a bulge here that base pairs to a loop that's located almost 4 kilobases downstream. And this has to form for frame shifting. So we don't need this. We're seeing good levels of frame shifting in SARS, but we still are wondering if long distance interactions might regulate, maybe reducing frame shift rates, maybe as polymerases and ribosomes moving up and down the RNA, the virus might want to increase or decrease frame shift rate depending on the stage of virus infection. And that can be done by refolding the RNA over long distances. My colleague Andy White has shown this beautifully with plant viruses, how the RNA refolds different long distance interactions to either make subgenomic RNAs or to do something like frame shifting or to replicate. So of course, because everybody is working on SARS-CoV-2 now, a lab did a really neat kind of a new technique that they call cross-linking of matched RNAs and deep sequencing. basically they cross-link all the RNA in an infected cell, so covalently attach RNA. So anything close to each other by base pairing will be linked covalently. And then they do deep sequencing, so they can sequence across these long distance interactions. And then they have to do some pretty serious bioinformatics. They map it all back. And what they found is that they found the frameshift element located here. So you can see the UUA, the shifty site, and that 3 helix pseudoknot here. But they found starting upstream and going through it, this big, long, they don't bother to show sequence on these dashed lines, but this huge, complicated, bulged out with some branches, stem loop region, and it comes all the way back around to base 845. So they call it the 1475 nucleotide frameshift element arch. So maybe all this long distance base pairing is affecting things. So now we have a hold of a clone that spans this region, and we're going to plug it into our dual luciferase reporters. I just want to quickly point out something that is actually one of the practical reasons why we care about the frameshift element structure is we, and again, a lot of others, think it's a good target for possible antiviral drugs because host mRNAs don't frameshift anywhere near this level, nor do they have this structure. So if you could find a small molecule that could bind in this complicated three-dimensional structure that inhibits or overstimulates frameshifting, you might block virus replication. So Jake Peterson and Walter Moss's lab modeled a 3D structure. So this is a 3D version predicted of our 2D structure shown on the left. And I sort of labeled the different domains so you can line things up. And when you have a 3D structure like this, you can get pockets. So it's not just alpha helix in an unstructured loop, or an A-form helix in an unstructured loop. You get all sorts of unpredictable things. So this program predicts deep pockets that are kind of open spaces where a small molecule might fit in. And then they used this big database of small molecules that are already FDA-approved drugs for working with humans. They found the ones that might fit into this pocket here. So there's actually one in there if you squint. And we just did our first experiments, really. Luke did this. So he added some drugs, and lo and behold, a couple of them gave a statistically significant reduction in the firefly luciferase activity and in frame shifting. But we can't get too excited because this could be causing all sorts of global effects on the cell. It could be affecting initiation as well. So we don't know the effect on translation or a lot of other things. But it's still kind of we're set up to start doing these types of assays now, is the point. So we have a proposal pending, and I'm really interested in the biology. So in all these structures that control frame shifting, and is it actually regulated over time? Do proteins bind? And then Walter Moss will be leading the screen for drugs we're basically going to pay the Broad Institute at MIT to do a big high throughput screen using the dual luciferase reporters. And then we can also look at virus replication, because at Iowa State now, Brian Belair has got a hold of the actual virus and has it up and running and replicating in the BSL-3 lab at the vet school. And we also got some full-length infectious clones, which is a cloning wonder. They did this so fast, this huge genome. And Pei-Yong Shi at University of Texas Medical Branch very readily sent us the full-length infectious clones, so we can mutate and look at the effect of these mutations actually in their natural context and how do they affect virus replication. So with that, I'll thank people in my lab, most of them who are doing it as a side project. They have other major things they have to do, but Wei Weizhou made all these clones, all these mutants, and more, a lot more than I showed. And Zach Lozier is getting going on in vitro translation and some of the bioinformatics. And Luke is a postdoc in Kathy Miller's lab. And then the guys, the really awesome RNA modelers in Walter Moss's lab. And I thank DPR for some support. And with that, I'll take questions if there's any time. Yes. Kind of sped up at the end. So how do we work questions?
So if anybody has questions, they can unmute themselves and just go ahead and ask you.
Yeah.
Hi, Alan.
Hi, Alan. That was awesome.
Okay, thanks, Roger. Yeah.
I guess the paranoia in me asked, and you touched on this at the end, how you guys handle this in the lab, and as all the infectious codes being handled out at BSL-3, or when when Wei makes all the mutants, how does he do that?
So we're working with the biggest clone. We're working with clones that are like 1% or 2% of the whole viral genome. That's all we have in my lab. I guess we have that. We have one that is about. We have that 1475 base sequence, and so that's all we have in the lab. So
that's not infectious.
So it's not going to be infectious because you need a 29,903 base genome to be infectious. So it'd be like if you took, it'd be like taking, we have a firefly luciferase gene. Are we going to have to worry about fireflies in the lab? And we have permits for it. We have a permit from the IBC here at Iowa State, and then Brian Belair has a permit, both of the federal permits and the local permits to work with.
So the infectious stuff is going on at the vet school?
Yeah, and it's a BSL-3 lab. They have to wear, the spacesuits and stuff. Yeah.
I just want to add one more comment. I posted this to Facebook yesterday, I think. A paper just came out in Cell. They did a genome-wide screen and found, I just read the abstract, found a bunch of molecules that either inhibited the COVID or helped it in infection. And you might want to take a look at that paper.
Yeah, there's a ton of those. I'm only looking at the ones that affect frame shifting. There's a whole lot of paper. Well, there's about 3 or 4 now that already have published or posted on bioRxiv drugs that are specific for frame shifting.
Oh, really?
And so, yeah, there's a lot. And I think we overlapped with one of them. But what we're doing is we're targeting certain pockets that and other people, and actually Walter has published with his competitor, sort of collaborator, competitor, a guy named Matt Disney. They found a drug that binds that attenuator stem loop, and it reduces frame shifting a little bit. And they already published in a ACS journal. So yeah, there's a lot. Big step though, from that to an actual drug that works in humans and doesn't have side effects.
Hi.
So Alan, this is Gwen. I just had a basic biology question on this. When the frame shift happens, it makes now the RNA-dependent RNA polymerase, but that is being made as a fusion protein with the protein that's upstream of it.
Yes.
So how does it must have to get cleaved in order to then be active, right? What cleaves it? Is it an external protease that's made even more upstream?
So that's what I pointed out when that polyprotein thing. Mm-hmm. So there's specific amino acid sequences that are cleavage sites all up and down that big protein. So yeah, and they are, I believe, they're all viral proteases for this virus, although it may not be known, but certainly the virus has at least 2 proteases that do those cleavages. So yeah, it gets cleaved, separated away from the other parts of the protein.
But you said before cleavage, some of these proteins, if they're cleaved at a slow rate, might have a different function than afterward. Yeah. Would this be one of those?
Could be. That happens with, there's a beautiful story with the toga viruses like chikungunya virus. So the polymerase, you start out with an uncleaved form and you make negative stranded RNA, the complementary to the viral genome, and then it cleaves more, and then the separate subunits can now make the positive strand RNA, more copies of that. So I don't know for coronaviruses though. I don't know if anybody knows, but yeah, it's possible.
Alan, this is Ido. Great talk. Couple of questions. One is, since you're working with partial genomes, how much would the structure of the RNA that you're working with be applicable to the total 3D structure of the whole genome? I guess you are focusing on a small part only, but you may be missing some long-range interaction. We talked a lot about those.
Yeah. So that's a good question. A woman, I think her name is Sophie Ruvkin, she's at MIT. She did this really awesome chemical modification of all the RNA in the genome that modifies only single-stranded regions, and then you do RNA-seq on everything. And the RNA-seq is affected by the chemical modification. So she basically found all possible known interactions, long distance, short distance, in SARS-CoV-2. And the Frameshift stuff really shows up. Though that's a really tight, that pseudoknot is really gonna form. But she did find the competing base pairing as well. So she could find alternative base pairings of the same sequence. And so, yeah, we're kind of, I think there's really, and then I described that other method, that comrades method. So yeah, we're definitely very aware of the long distance interactions. But I think that pseudoknot for sure forms and does cause frame shifting in the full length context, as well as our little short context.
So my second question is a more bioinformatic tool oriented one, which is, you said that tools for finding pseudoknots are not very good. Is there a market for that, developing something that identifies pseudoknots?
It's what is it? It's a NP hard problem.
Well, NP hard problems are not problems if you can solve them with heuristics. The question is, I mean, I've seen all sorts of software that purport to solve sequence alignment is NP hard, and we do it all the time, right?
Okay.
That I've seen all sorts of software that purport to solve pseudoknots. And, and you know, I don't know. I've never delved into it. I don't know the level of accuracy and so on. Is that? And you said that it's a problem that you need better.
Yeah, I mean, computationally, the programs that most people still use, they'll basically fold the RNA and then take the single-stranded regions and look to see if they can base pair to find pseudoknots. And there's also just plain rules of RNA folding. I think pseudoknots are more stable than you would predict if you just use the rules of the energy of the Turner rules, which are how stable is a CG pair, and then how do the flanking base pairs affect that stability. So yeah, I was thinking, I don't know if anybody has used, somebody probably has. I was thinking it'd be an interesting challenge for, you know, machine learning or something. Just feed it all the known structures and have it find its own rules. But it's probably been done. Who knows?
Thanks.
Sure.
Any other questions for Alan?
Alan, so sorry, I had to, great talk, but I missed, unfortunately, most of it because I had another meeting.
It's been recorded, I think. It's been
I know, I saw that. I'm going to listen to the whole thing.
Okay.
And you may have addressed this, but one of the things we noticed looking at some of the RNA-seq data is that all the transcripts of all the ORFs are translated sort of more or less in tandem in patients with and without COVID, but the 1A has a completely different pattern than the rest of them. Is that?
Oh, that's interesting. Well, the very first protein, let me see if I can go up here. I wanted to talk about this. I could give a whole stuff just on translation. If we go to this slide, are you still seeing my slides?
Yeah.
Yeah, okay. I guess I'm not in presentation mode.
Yeah, change your mode because we only see part of it.
Okay, well, you can see the important
Yeah,
Right.
great, thanks.
part. Right. So this NSP1, which is part of ORF1A,
Uh-huh.
that protein shuts down a lot of host translation. And there was already a paper in Science with all the cryo-EM data showing how it binds the ribosome, and also all the, like, they had a whole virology medical aspect. So it shuts down translation of most genes by interacting with the leader sequence, but it especially hits the interferon stimulated genes, so the host defense response. And somehow all the viral RNAs, because they all have this leader sequence, that black sequence, that sequence somehow allows RNAs to be translated in the presence of NSP. So it allows them to escape the inhibition. And I just saw a paper about pyrimidine rich, something about the nature of the genes that kind of are not as inhibited as much by NSP1. But I could easily see how that could affect an alteration of the translation of the ORF1A, just that protein alone could really affect a response, host response to infection.
Okay, thanks.
Sure. And I'm interested in that paper, by the way, yeah. Or whatever you found, your findings. Yeah, we should.
Okay, absolutely.
Yeah.
I'm giving a talk tomorrow at 4, I think. What time?
Okay.
You
I was
know,
just gonna say, I thought I missed your talk. Good. All right.
At 4:20, Eve.
Okay. at 4:20.
Okay. Okay. Good. So more on SARS-CoV-2 and COVID, everybody, tomorrow at 4:20. Is that a B?
So it's the BCB 691. It's a student seminar.
Oh.
Unfortunately, I won't be. I'm actually the faculty who's supposed to host this, but I have a conflict. But I bet Eve can take care of herself. So if anybody's interested in hearing Eve's talk, just email me or her with a request for the Zoom link.
Okay.
If Eve's comfortable with that, that is.
Oh yeah, I don't, you know.
Okay.
All right, well,
It's a student seminar, but there's no reason if the faculty are good with it, they can't, show up and hear. If that's okay.
okay, since we're a little over time already, I'm gonna officially end seminar so people can be free to leave. But if anybody wants to stay online and Alan's available, feel free to stay and ask questions after close. So thanks everybody for being here and attending, and we'll see you next week. And again, if anybody want to stay and talk to Alan, I'll leave the meeting open. And thanks, Alan. I guess we'll do our little silent clap or
Oh yeah, thank you. Thanks.
symbol. Thanks.
Thanks, Ellen.
All right.
Let's see.
Now I'm gonna move over.
I'm going to sign off.